welcome to Biology, the scientific study of life. Primary production in marine and freshwater ecosystems. In aquatic, marine, and freshwater ecosystems, both light and nutrients are important in controlling primary production. Light limitation. Because solar radiation drives photosynthesis, you might expect that light is a key variable in controlling primary production in oceans. Indeed, the depth of light penetration affects primary production through the photic zone of an ocean or lake. More than half of the solar radiation is absorbed in the first meter of water. Even in clear water, or only 5 to 10% of the radiation may reach in depth of 20 meters. If light is the main variable limiting primary production in the ocean, we would expect production to increase along a gradient from the poles toward the equator, which receives the greatest intensity of light. However, you can see, notice back to satellite images, you can see that there is no such gradient. Some parts of the tropics and subtropics are very unproductive, while some light latitude ocean regions are relatively productive. Another factor must influence primary production in the ocean: nutrient limitation. More than light, nutrients limit primary production both in different geographic regions of the ocean and in lakes. A limited nutrients is the element that must be added in order for production to increase in a particular area. The nutrient most often limiting marine production is either nitrogen or phosphorus. Concentrations of these nutrients are very low in the photic zone where phytoplankton live. Ironically, they are more abundant in deeper water where it is too dark for photosynthesis. As detailed here in this figure, notice that nutrient enrichment experiments confirmed that nitrogen was limiting phytoplankton growth of the south shore of Long Island, New York. Practical applications of this work include preventing algal blooms caused by pollution that fertilizes the phytoplankton, eliminating phosphate from sewage will not help solve the problem of algal blooms unless nitrogen pollution is also controlled. Several large areas of the ocean, however, have low phytoplankton densities in spite of relatively high nitrogen concentrations. For example, the waters of Sargasso Sea, a subtropical region of the Atlantic Ocean, are among the most transparent in the world because of their very low density of phytoplankton. A series of nutrient enrichment experiments revealed that in this case, it is availability of the micronutrient iron that limits primary production, as described in this table. Such evidence that iron limits production in some oceanic ecosystems encourage marine ecologists to mount several large-scale field experiments in the Pacific Ocean during the last 2 decades. For example, in one study, researchers breed low concentrations of dissolved iron over 72 kilometers square of ocean and then measured the change in phytoplankton density over a 7 days period. A massive phytoplankton bloom occurred as indicated by a 27-fold increase in chlorophyll concentration in water samples from the test sites. Why are iron concentration naturally low in certain oceanic regions? Wind-blown dust from the land is the main process delivering iron to the ocean, and relatively little dust reaches the Central Pacific and Central Atlantic Oceans. The iron factor in marine ecosystems is actually related to the nitrogen factor. Where iron is limiting, adding iron stimulates growth of cyanobacteria that fix nitrogen, converting atmospheric into nitrogenous compounds. These nitrogenous nutrients in turn stimulate proliferation of eukaryotic phytoplankton. This relationship can be summarized as this. Notice that iron positive cyanobacteria plus nitrogen fixation, phytoplankton production. Notice also the inquiry which nutrients limits phytoplankton production along the coast of Long Island. Pollution from duck farms concentrated near Morch Bay adds both nitrogen and phosphorus to the coastal water of Long Island. Researchers cultured the phytoplankton the results here. Notice the results also. Conclusion: since adding phosphorus, which was already in rich supply, had no effect on growth in island, whereas adding nitrogen increased algal density dramatically, researchers concluded that nitrogen was the nutrient limiting phytoplankton growth in this ecosystem. Notice also the experimental eutrophication of lake. Notice eutrophication from the Greek eutrophs, nourished. Areas of upwelling where nutrient-rich deep water circulate to the ocean surface have exceptionally high primary production, which supports the hypothesis that nutrient availability determines marine primary production, because the steady supply of nutrients stimulates growth of the phytoplankton populations that form the base of marine food webs. Upwelling areas are prime fishing locations. The largest areas of upwelling occur in the Southern Ocean, Antarctic Ocean, and the coastal waters of Peru, California, and parts of Western Africa. Nutrients limitation is also common in freshwater lakes. During in 1970, scientists including David Schinder from the University of Alberta, Canada, noted that sewage and fertilizer runoff from farms and years added large amounts of nutrients to lakes. In many lakes, phytoplankton communities that had been dominated by diatoms or green algae became dominated by cyanobacteria. This process, known as eutrophication, has a wide range of ecological impacts, including the eventual loss of all but the most tolerant fish species from the lakes. Controlling eutrophication requires knowledge of knowing with which polluting nutrients enables the cyanobacteria to bloom. Nitrogen is rarely the limiting factor for primary production in lakes. The scientific notice David also conducted a series of whole lake experiments that point to phosphorus as the nutrient that limited cyanobacterial growth. His research led to the use of phosphate-free detergents and other water quality reforms. Notice the lake, the experiment eutrophication of lake. In 1974, the far basin of this lake was separated from the near base. Notice by plastic cureton and fertilized with inorganic sources of carbon.